because we are looking at something, and because the hematopoietic stem cells represent a very, very small fraction of the total mononuclear cell fraction in here, the data files that we have to collect to do it are very large. As we talk, as I think Laura went over with you in the other the other lecture, talking about rare events analysis and things like that. So in order to be able to truly identify the cells, you'll see here that even in you have your compensation controls, which are here. The top four here, these are your data files. The data files have different numbers of events that are collected just because of the size of the sample and stuff that we can work with. So that here, your top one actually has 10 million events that's been collected in there. That's why that thing was so large and it took you so long to download it. Just because there's the sheer number of events in there. So that's one thing to get used to, is that you're going to be working with huge files. Even these two are about 6 million. Well, this one's almost 7 million, and then this one has 2 million. The smallest one has over 2 million events that were collected in it. Then what you have is you have all your compensation controls, which are right here, including your unstained here. Now, this was where the other class got a little bit confused. As we were talking about this. In terms of, on this panel here, which we've done, this is like essentially the experimental setup that I put together when we were doing this experiment. And what it's done is it's labeled in things like lineage FITC, CD45, APC, and things like that. So I've actually labeled it according to the color of the antibody that's been used. However, on the machine, the way the machine does it is according to the laser and then the filter set, which is then used. So, does everybody remember all about the filter sets and all about the lasers and everything like that? Okay, the other class, nobody had a clue. So to try and make it a little bit simpler here, again, this is just the Excel spreadsheet that I used when we were setting up this experiment. And what it does is it was kind of like the cheat way or the easy way of showing according to the laser and the filter right here. This is the different things that we use to detect it, and then this is the fluorochrome or the colors that we use. I can send this to you. Unfortunately, I don't have access to Blackboard for this class. Pardon me, I don't mean it. But yeah, I can email this to you guys. I can email it to one person, and I don't know, something. Or just
picture if anybody wants me to send it to them.
There you go. So basically what it is, so if the first parameter that you were going to look at was lineage FITC, which is this one, it's actually the 488 laser with the 52550 filter set. You're going to be talking, we're going to be discussing here all of these different antibodies. We're going to be discussing all of these different targets. What we talked about in terms of CD34, CD45, 45RA, CD98, CD38, all of these. This kind of shows you, because when you go into the machine and into the actual analysis of it, it doesn't give you the color that is used, although that's what I've written down. It actually gives you the filter set and the laser that's used. So when you start asking what does 488, 610, 20 mean, refer back to this, it'll start giving you different colors. Make sense? Okay, good. So, the first thing, yeah. The first thing you can do, or you should do, is actually go through and compensate this. Did you guys did all the compensation stuff before in your other practical and your other assignments? So The compensation, although I was told by the postdoc who actually collected this that the data is pretty well compensated because she compensated it when she collected the data, you may need to just go into the compensation matrix and double check it all again. So I would, you guys have any questions about the compensation? Lots of questions about the compensation. All right, let's see. So with the compensation, now this is different because the newer version, I'm not totally familiar with this. But within your samples, you have these, you have all the data, all the samples, and then you have those samples which have been designated the compensation controls, which are for all the different parameters which have been collected, as well as the unstained control. If you go into your compensation matrix like this, it will calculate it like it is, like you've done for your other assignments, yeah? Now, the problem is, with this, is that in certain cases like this, what it does is it automatically goes in and gates upon a population of cells down here. Now, sometimes these are not exactly what you'd want them to be because they're missing out a whole series of populations of cells and things like that. So then what you need to do is in this compensation part is actually then go in and try and adjust the different cell types, the different populations of cells here.
We just have to cover the items divisions, right?
Yeah, yeah.
So are we supposed to gate everything?
Then from this,
We do that to all of them, right?
yeah, you're probably going to have to go and do it to all of them.
So we gate all of the cells in the picture? Or-
Sorry?
So we gate all of the cells, like in that first one on the left? It's every, like the whole thing?
Yeah,
Or
here.
just-
So you want to gate on all of the cells here. What this is doing is the machine is sort of gating on just something that's very small down here, which seems to be debris. Down here, most of the cells are here. And it's only once you gate on that do you then actually see the correct compensation and the gating on the positive population. So what you need to do is actually go through all of the different samples.
The one you said was debris, how did you know that that was debris?
Just simply based upon size. You know, when we set the machine up.
Okay.
So this is very similar to what you did in your previous assignments. The difference here is that you're going to have to go through almost all, you're going to have to go through all of them and do it. You have multiple different parameters. So you go through that, that'll set this up, and then of course it'll calculate out the compensation matrix that you can then apply to your different samples. So, I'm going to pretend that we did that. The next thing that we're going to do is we then need to start gating on the population of cells and gating on the hierarchy. So, if you have problems with the compensation, like I say, the data itself is fairly well compensated. The postdoc who did it said that she thinks she did a pretty good job compensating it, so it should be okay. But you should probably just go in and check it. So the first thing that we need to do, and then this is what you need to do in this, is that we need to start building up the hierarchy of how do we actually identify the different subtypes and how do we identify the hematopoietic stem cells. So, according to this, if you guys understand, what is the first step that we need to do? And it's usually the first step that you do with any data.
Gate.
Yeah.
Single cell?
You gate according to the size parameters of forward scatter and side scatter. Like this. So this is what we have here. So the first step is to create a plot that looks like that, which is forward scatter and side scatter. Yes?
Stupid question.
There's no stupid questions, no stupid answers.
What is the difference? Like, what do they measure? Like, what's the difference in the measure?
Between forward and side scatter? So just the simple, oversimplified explanation of it is that if you have your cell, which is here, and then you have your laser right here, shining out the laser, what happens is that you have two detectors here. So this one is your detector for forward scatter. This one here is your detector for side scatter. When the laser shines on this cell that's passing through, what happens is that the laser light passes over it, around it like this. So what forward scatter, you can think of it as a simple measure of size. It's how light passes, it's how that cell kind of blocks the light while it's detected. Within the cell, so this laser is also shining in here, within the cell are various intracellular granules, lysozymes, peroxides, whatever. I mean, just various intracellular granules. What happens is the laser then hits those and gets reflected off at an angle. That is what side scatter is. So side scatter, you can think of as a measure of granularity of the cell. What it is just a physical, it's a non-fluorescence based, it's just a physical description of the cell type. So what happens is that if you think of this right here, anything that has, so if this has been set up correctly in terms of like the voltages and things like that, you would say here that different cells in the hematopoietic system have a different position based upon just their characteristics. So if you were to think, okay, you guys know all the different cells of the hematopoietic system, like for example, lymphocytes. Lymphocytes have a large nucleus, they don't have very much cytoplasm, and they're small and they're round. So what happens is you would expect down here, because if this is forward scatter, this is size, this is granularity, they have very low granularity, they're actually fairly small in size, so your lymphocyte population would be down here. If you were to look at something that was, say, a lot larger and had a lot more granules, so granulocytes and monocytes, you would find them over here. If you were going to look at something that was maybe also granules that came from granulocytes but wasn't quite as big, such as neutrophils, so this would be monocytes here. Right here, you would find neutrophils. And then over here, you could also find lymphocytes that have been blasted out. So lymphocytes that have been activated and they were bigger, but they don't have the level of granularity. So based upon the size markers or the size characteristics of the hematopoietic cells, you can kind of say, okay, well I know that this is probably lymphocytes down here. This is probably a blast lymphocyte population. Here you might have some, well this is a